Hello everybody, my name is Vitor. I'm going to present my master's project entitled The Role of NALP1 in the Response of TH17 Lymphocytes. My supervisor is Professor Nils Olsen at the Department of Immunology. I brief introduction to T cells. I work with TH17 and Treg cells. TH17 are very pro-inflammatory cells, while Tregs are very anti-inflammatory T cells. The main marker of TH17 is the Targhetta transcriptional factor, while the FOXP3 for the Treg cells. There is a very interesting feature of the cells that they can almost transdifferentiate themselves into each other. So, and this mechanism is present in a lot of biological processes as autoimmune disease, allograft rejection, and so on. So I also mentioned inflammasomes in the title. Inflammasomes are complex platforms of proteins responsible for the activation of pro-inflammatory cytokines, IL-beta and IL-18, for example, and they can induce cell death via periapoptosis. So the aim of this project is to verify the importance of NALP1 in TH17 cells. First, I will show a result of the immunophenotype that we did in mice lacking the NALP1 and in comparison with wild-type mice. So we look at the main lymphoid tissues, thymus, spleen, and lymph nodes, and the only significant result that we got was that the percentage of FOXP3 cells was slightly decreasing in NALP1 KO mice in comparison with wild-type ones in the spleen. So we also try to look at the influence of Valborapro in the activation of Treg and TH17 cells. Valborapro is a potent activator of NALP1 inflammasome, and for that we isolated TH17 and Treg cells, and we look at their respective markers, Targhetta and FOXP3. So the big difference that we saw here is that in the Treg population, upon treatment with the Valborapro NALP1 activator, we saw a big decrease of the percentage of FOXP3 positive cells, as well as the expression of the FOXP3 measured by MFI. So we saw that in all of the concentrations used, and interestingly, this seems to be independent of NALP1, since the NALP1 KO mice show the same behavior. So in conclusion, there is no big difference between wild-type and NALP1 KO mice. T cells treated with Valborapro do not undergo periapoptosis, and Valborapro is capable of reducing the percentage and the expression of FOXP3 in Treg cells, and this is independent of the NALP1. So thank you. I would like to thank all the lab mates and Professor Nils for the opportunity.